Good.
Thank you, everyone. My name is Ellen George, and I'm a PhD candidate at Cornell University with Lars Rudstam and Matt Hare. And today I'm going to be talking about some of the genetic work we've been doing with Cisco and Lake Ontario for the past few years. I'm basically trying to condense almost an entire PhD in 15 minutes, so we're going to go quickly. For those of you that have been in the room for the last few talks, you already know a lot about Cisco and Lake Ontario. What's important for this talk to know is that historically Cisco used to spawn all over Lake Ontario, both the western and eastern sides. And that's what this map is. Every red star is where a spawning population was documented before the population collapsed in the early 1900s. Today, however, after that collapse in the late 1800s, early 1900s, we now have two known spawning populations that we have been studying. One is on the New York side in Chameau Bay, and one is on the Canadian side in the Bay of Quinte. So as interest in restoration of Cisco across the Great Lakes, but especially in Lake Ontario, has been ramping up in the last few years, we realized that not only do we not know a lot about these populations in general, but we especially don't know a lot about, or anything about, the genetic status of this population. So there are a few genetic concerns that we wanted to address with this study. The first is we know that the Cisco and Lake Ontario went through this huge population decline about 100 years ago. And along with that population bottleneck, we don't know if they also experienced a genetic bottleneck, which is a huge loss of genetic diversity following this population decline. So our Cisco today still suffering the effects of low genetic diversity from this bottleneck. We also don't really know that much about the stock structure in the lake. So we know there's these two major spawning areas, Chameau and the Bay of Quinte, but to what degree these stocks are mixing, or how reproductively isolated they are, we don't really know. And finally, we are also concerned about the rate of hybridization with lake whitefish. Lake whitefish and Cisco can hybridize and do hybridize in the wild sometime. These hybrids are sometimes caught in the Bay of Quinte by commercial fishermen, and they call them Cisco mules. So we are concerned about how, if this is a high rate of hybridization, how that's going to impact the recovery of Cisco in the future. So to address these genetic concerns, we collected different tissue samples from several different aggregations around Lake Ontario. The Canadian side, we got, all the samples from the Canadian side, we got scale samples from the OMNR. And on the American side, it was all thin tissue in ethanol. So two different tissue types for this study. We definitely collected lots of samples from the two different spawning populations. So we have samples from 2015 and 2016 from Chameau Bay. And then on the Bay of Quinte side, we got actually a really nice time series from between 2003 to 2017 of different spawning fish from those years in that time series. Something else that was really cool that we were able to get from OMNR is some historical scale samples from Wellington, Ontario, which is, if you saw Taylor's talk, is close by those Brighton sites where they were collecting larvae. So these are from 1980. This population hasn't really been sampled as recently as much, so we don't know if this was maybe a spawning population that was still active in the 80s but is gone now, or it may still be a remnant population now. We're not sure. So but we included those samples in this study. Also, we were lucky to get some tissue samples from some fish that were caught in the summer during midwater trawl sampling with USGS. And these fish, because they were caught in the summer, they don't represent spawning populations like the Chameau samples, the Quinte samples, and maybe the Wellington samples do, but they could potentially be a mixed stock once the fish leave the spawning area and are spending the summer out in the cold waters of Lake Ontario. These could be fish that represent multiple stocks, or even stocks that we don't know exist yet. So, so those are some other types of samples that we could potentially be looking at. So to look at all of these different genetic measures that I'm going to share with you, we used 8 different microsatellite markers that amplify in both lake whitefish and Cisco. and have been used in these species across the Great Lakes pretty extensively by Wendy Stott at USGS in Ann Arbor. So these are markers that, while they amplify in both species, some of the markers show pretty different allele frequencies between the two species as well. So some are a little bit more similar, some are different, but they amplify in both species, so we can use all 8 markers. So first I want to share with you some structure plots that show the different stock structures in Lake Ontario. These plots are created by putting our microsatellite data through a program called Structure, which is a Bayesian clustering method that assigns each individual fish to a certain population. So this is all of our data, including both all the Cisco populations and the lake whitefish as well. In this case, Structure said that it's most likely that we have 2 populations, both the different species. So they're different species. But this is, I like this graph because it really shows you, one, that we are dealing with different species, but also some kind of cool things to do with hybrids. So in this plot, each individual is represented as a vertical bar, and each, what we're calling population, so sampling location and year, is split up into different blocks. All the green are Cisco on the left. Way over on the right are lake whitefish in red. But in the middle, you can see a few vertical bars that are half and half. So half green, half red. And these represent potential lake whitefish Cisco hybrids that were caught during sampling. So I'll talk a little bit more about those later. But anyway, while we're looking at these 2 different species, I also wanted to share the FST value with you. So this is a measure of genetic distance between the 2 species. How different are these species genetically? And our FST value for between 2 different species is 0.21. So I want you to remember that number just for one more slide. Don't worry, not too much longer. Okay, but of course we're interested in stock structure between just the Cisco. So I have, in this plot, I've taken lake whitefish out, and we're just looking at the different Cisco populations in Lake Ontario. So in this case, running this data through the structure program showed that a single population has the highest likelihood. So we actually don't see any evidence with these markers of any stock structure in Lake Ontario. The k = 1, or the 1 population graph, is very boring. This is just a solid block of color. So I've shown the k = 2 plot here because it's more interesting to look at. And also just to show that there really is not a lot of difference between the different populations here. They all have pretty similar allele frequencies between the 2. The one population that does look a little different is that Wellington sample, which is the historical 1980 samples from Ontario. However, those were also scale samples that were pretty old, so it had a pretty high genotyping error, so we think that that's just a relic of the fact that we had a lot of allelic dropout and issues reading genotypes with that population. But even with those differences, it still was not significantly different than the other populations in Lake Ontario. So at least genetically, no sign of any stock structure in the Lake Ontario populations. The 2 spawning populations are pretty similar genetically. Just to drive that point home, if you remember the FST value between 2 species is 0.21. The average FST value between all these Cisco populations is very small, just 0.03. So not a lot of genetic differentiation between these subpopulations. Okay, so if we now know that there's not very strong, or there's no evidence of genetic stock structure in Lake Ontario, let's dive a little bit into the diversity measures and look at how diverse each of these subpopulations are. So if we combine all those Cisco populations and look at their genetic diversity, we found that we had really high allelic richness, which is the number of alleles per the 8 different loci that we looked at. Pretty high number of alleles across each population. Also, putting our data through program bottleneck showed that we didn't have any significant excesses of heterozygosity in any of these populations. And what that tells us is that we're not seeing any evidence for a strong genetic bottleneck for any of for the Cisco population in Lake Ontario. So even though they went through this really big population decline about 100 years ago, it doesn't seem to have really strongly affected their genetic diversity. So they've managed to maintain pretty high genetic diversity despite this big population decline. Also, if we look at the individual populations, they have very low FIS, which is also called the inbreeding statistic. It's a measure of genetic diversity within each subpopulation. They have very low inbreeding statistics for all the different populations we looked at. Also very high expected heterozygosity across all the different populations. So what that tells us is not only are these Cisco and Lake Ontario pretty genetically diverse, all the different populations have a similar level of diversity. So it's not just one population, for example, that for some reason is experiencing really high inbreeding or low genetic diversity. It seems to be high diversity across the board. We also looked at the effective population size, which is abbreviated NE, and effective population size is the size of a quote-unquote ideal model population that would be experiencing the same rate of genetic drift or loss of genetic variation as the population of interest that we're looking at. So in fish populations and marine populations, they often have census sizes in hundreds of thousands, millions, whatever, but the effective population size can be much, much lower. And a low NE is bad news. So you do not want a low effective population size. If you have a low effective population size, it means that your population is exhibiting genetic drift or random genetic changes at a much higher rate than you would expect given the census size of the population. And what that means is that this population is at risk for a higher rate of loss of genetic variation than you would expect given that you have what looks like a big population of fish. So we combined all of the Cisco populations for Lake Ontario except for those historical Wellington samples because we wanted to get an idea of the current effective population size. And we found actually a pretty high effective population size. So NE of about 5,000 with an upper bound of infinity. Infinity Cisco. So, yes, my dream. So that's really good news. That means that it doesn't look like this population is at risk for losing a lot of genetic diversity based on random changes in allele frequency. Okay, so I mentioned those hybrids in that structure plot, those little bars that were half red and half green. We were concerned about the rate of hybridization between Cisco and whitefish for several different reasons. First of all, lake whitefish and Cisco, at least in Chameau Bay, are often spawning on the same substrate, but they are temporally segregated by about 2 weeks. So often the lake whitefish will come in and spawn and leave, and then about 2 weeks later the Cisco come in and spawn and leave. That's in a normal year. In a couple of the last few years, though, we've had some weird falls where it's been pretty warm late into fall, and then all of a sudden it's cooled down very quickly near the end of fall. And when that has happened, it seems like the whitefish and Cisco are coming in at the same time and spawning. So we'll catch a ripe Cisco and ripe whitefish in the trap nets on the shoals at the same time. And they can hybridize, at least in the lab, and sometimes in the wild they've been observed hybridizing as well. So we were concerned about that. We also, Taylor mentioned earlier that we have some weird larvae in Lake Ontario. The majority of our larvae cannot be keyed out to lake whitefish or Cisco. Instead, they show these intermediate overlapping characteristics. So we were concerned that that meant that there was such a high level of hybridization that we were getting all these hybrid larvae. And also, we do know that fishermen have reported Cisco, what they call Cisco mules in the Bay of Quinte, which are adults that display intermediate characteristics between lake whitefish and Cisco. And so we were concerned about how, if there was a high rate of hybridization, how is that going to affect Cisco restoration in the future? And so in order to identify wild hybrids, we developed a new panel of 3 different markers, genetic markers that can identify a wild hybrid. 2 of the markers are fixed in the 2 different species, so Cisco will always show one banding pattern, and lake whitefish will always show the other. One of our markers is not completely fixed, but it shows a very different allele frequency between the 2 species. For example, just to show an example of how this works, up here is an image of a gel. On the left, in yellow, there are 3 lake whitefish individuals, and they have a smaller a smaller band size, and so it travels farther down on the gel. On the right, we have 4 Cisco species in red, and they have a larger band size, so it doesn't travel as far on the gel. The fish in the middle, who are in blue, are all hybrids. So they display both the lake whitefish band and the Cisco band. So it's a pretty clear way to see if you have a hybrid or not. So using these three marker panel, we were able to identify 11 adult hybrids that were collected in the Bay of Quinte. These were fish that were collected during the commercial fishing season and were identified upon collection as having intermediate characteristics between Cisco and whitefish. One thing that was really cool and really interesting about them, though, is that 3 of these fish, who were hybrids, showed banding patterns that suggested that they might be F2 back crosses, which means that one of their parents was a hybrid and one of their parents was either a full-blooded Cisco or whitefish, which means that if these are F2 back crosses, that a hybrid successfully came back and spawned and back crossed with either a Cisco or a whitefish. Which is not what we were expecting, because a lot of times the commercial fishermen will report that they have these mules, but they won't be ripe, they won't have developed eggs, or they appear to not have developed eggs in the past. However, with these F2 back crosses, it shows that at least some hybrids are able to come back and are ripe and are able to spawn and create viable offspring. And this was further supported by the fact that in Chameau Bay in 2017, we collected an adult female who was ripe with eggs, and she was genetically identified as being a hybrid. So it seems that while at least some of these hybrids, although hybrids do survive to adulthood, at least some of them are ripe and may be able to back cross and spawn with Cisco and whitefish. What this doesn't tell us, though, is the rate of hybridization. So these are confirmations of adults, but we don't get a rate from this. So to look at what the rate of hybridization might be, we ran these 3 marker panel on over 700 larvae that were collected from Chameau Bay in 2014. And we did find larval hybrids, but the rate was very low. 0.1% of the larvae were identified as hybrids. So low, but not unheard of. One nice thing that this means is that the huge amount of overlap in larval morphometrics that we see is not due to the fact that we have all these hybrids in Chameau Bay. We just have very weird larvae that are hard to ID. So hence the genetic